Okay, we are in chapter 5. Now, our next test is not over the 2 chapters. And chapter 5 is genetics, and we do Mendelian genetics, quite a bit of terminology in it, and then chapter 37, which we have a set of notes for that, which should be in your set of notes, and that's what we'll cover out of that chapter. We won't even go to it in the book. But starting here in the genetics chapter, hopefully, how many's had a little bit of genetics in high school? Done a few squares, put in a few stuff. Okay. We'll start simple, we'll add to it a little bit. Let me give you a little background on it. Who's the father of genetics? Gregor Mendel. He's the father of genetics. And they'll tell you in the book that he was good at what he did. He had a degree, although he never passed what was called the exit exam for this degree. But he did get a degree, and I don't remember what institution he got it from, but he was a monk, lived in a monastery, had a lot of time on his hands, obviously, because he chose to do this project. He actually developed what's called a science club, where he had several neighbors, farmers, different people in the science club, and they all picked different kind of projects and reported back from time to time on what was going on. Well, he chose genetics. Nobody ever chose that before, but he did. And he worked with pea plants. We work with fruit flies now. It's one of the main things we work with, but he worked with pea plants. And one of the things that made him so successful is that he worked with one thing at a time, and he wrote every little thing down. He didn't miss a dot. I mean, he wrote it down. And by documentation of everything, he knew what these individual things did. So once he figured this one out, then he'd figure this one out, then he'd add them together, see what happened. And, you know, when he got into things, it stumped him. And we'll talk about intermediate inheritance, where in 4 o'clock flowers, he had white flowers and red flowers, and he wondered which one's dominant. So he mixed the two, and he didn't get either one of them dominant. He got pink flowers. So that one stumped him, because that wasn't anything like he had done before with pea plants. So and he worked some of those out. But unfortunately, as he finished his career and tried to give his genetic information to other scientists where they could continue, they weren't interested. So he made sure it got put in some libraries, and by doing so, later, several years down the road, they found his work, and that's when they named him Father of Genetics. It's like 16 or 18 years after he was dead is when they figured out this guy was pretty sharp. They may have changed a few terms. I think he used the term factor, and now they use the term allele. But other than that, he's still known as Father of Genetics. Now, there is some basic terms that we have to understand to get a grip on genetics. If you understand these basic terms, you can read a question about genetics and know what to do with it. If you don't understand the terms, then you don't even know where to start. So it's important that we grasp these terms. The term homozygous. Anybody ever heard that term before? This term is in reference to something pure. I read this stuff in books all the time. Genetics is a big thing in the cattle industry. And, you know, you have different breeds of cattle. Some of these breeds have red ones. Take Limousine, for instance. Simmental, there's red ones and there's black ones. Well, you can have a black Simmental bull that carries a red gene. But they actually can do DNA tests on this bull and determine if they carry a red gene or not. And so if they don't carry a red gene, they would be listed as homozygous black. They have no red gene. Same thing with horns. You can be polled, which means there's no horn gene there. So there's a lot of breeds with different colors and stuff, but they can test for those colors. And in Simmental, there's also diluter genes that causes spotting or streaking, and that's not necessarily a trait that we look for in there, but they can identify the diluter gene that causes that. If they don't have the diluter gene, then they would be pure. So homozygous means pure or the same. Now in Mendel's work, if he represented T for tall, big T is dominant to little t, which is dwarf or short, in his green plants, homozygous would be in reference to big T, both alleles being the same, or little t. As long as those are the same, it's homozygous. Heterozygous means different. Heterozygous would be big T, little t. The alleles are different. That'd be crossing a Hereford and an Angus. That's a heterozygous cross, okay? Genotype is the actual genetic makeup. Can you see all the genotype of an animal or an individual? No. You cannot see that genotype. Can you see the phenotype?
Yes.
Yes. The physical makeup is what you can actually see and describe. That's your phenotype. Genotype is possibly what you can see. There's quite a bit of it that you can see, but there's traits there you cannot see. And I think a good example of this is when we're dealing with sex-linked traits. And in color blindness, for a female to be colorblind, she has to carry the colorblind trait on both X chromosomes. That's why there's very few colorblind females, 1% of the population. 5% of males, because if a male having 1X has a colorblind trait on it, they're colorblind. So here's a great case. If we take that away and say there's a normal gene there for her, she sees normal. But do you know by looking at her and by her describing colors that she's a carrier for color blindness? You cannot see that, can you? Now what'll happen is she'll marry this colorblind guy, probably feeling sorry for the way he dresses. And their first child will be this boy down here. And he's colorblind. Now we know for a fact Dad's colorblind, it's because he dresses pathetic. Who gets the blame for producing that colorblind child?
Mom, because she's carrying the other gene.
That's because y'all are educated. If y'all didn't, the average person out there, not being educated, since y'all got a little background in genetics, would say, Dad's colorblind, way to go, Dad, you produced another colorblind son. Blame it on Dad, right? Because the obvious, Dad's colorblind, son's colorblind. What did Dad give his son? Gave the Y, right? Dad didn't do it. Mom's got a genotype that we did not see physically, but it was expressed in her offspring. So you always get your bad traits from your mother. I'll come back to that. I'll have to prove that a little more, but I'll get it done. All right, so there's your genotype and phenotype. And they'll talk about that quite a bit, but they'll sure enough use these two terms. In a problem, I gave y'all some problems. Everybody get one of those? Everybody get that handout I was scattering around? Okay. And you probably reference that more next week than this week. But in some of those problems, they'll say, brown eyes are dominant to blue eyes. A homozygous brown-eyed person. What does that tell you?
They carry
Homozygous, if brown eyes is dominant to blue, and we have a homozygous brown-eyed person, it's two big B's, right? So if they say a heterozygous brown-eyed person, big B, little b. If they don't say anything and say a blue-eyed person, we know it's recessive, so the only way we can get blue eyes is to have two little b's, because there's not a dominant trait to cover up the weak trait. lost anybody? With me so far? All right.
I have a question.
All right.
Can two blue-eyed people produce a brown-eyed child?
Yeah, in eye color, probably, because we set up here and we use these simplified problems that brown eyes are dominant to blue, and we work these problems out. But in all reality, there's 8 different genes involved in eye color. And with 8 different genes, you can't see 2 brown-eyed people and they produce a blue-eyed child and look at mom and say, whose kid is that really? You really can't, because you could get you could have 5 kids and have 5 different colors. We don't know what's in that gene pool back there, really.
We were taught many moons ago that it was impossible, and that caused quite a bit of distress for
Oh,
people in
yeah.
our class.
Eye color, you can't go by that, because there's 8 different genes that's involved in producing eye color. And then you get those really odd ones where somebody's got a brown eye and a blue eye, or part brown and part blue in one eye. You ever seen your own eyes look brown one day and hazel or greenish the next? What? Explain that one. Maybe you're a little fickle and your body can't make up their mind what they want to do with it. You wear something green, your eyes look more green. You wear something brown, and some people just have brown eyes because they're full of crap. So, yeah, you can't, a lot of our simple problems you can't really go by, because there's polygenic inheritance involved in eye color. It means there's many genes involved in it. We don't want to even get into that at all.
We were just taught like 2 recessive genes could not produce a dominant trait, or 2 recessive traits could produce. It's blue and
Yeah.
recessive.
That's just messed up. There's something else we might do in here. It won't be next week, maybe the following week, if we have time. I've got a little, and just having one Zoe class, sometimes I do this. I have one little exercise that we can do. It takes a little while to do it, but since it looks like we're equivalent, we're matched up in here. What we do is 2 people start going through this exercise, and there's I want to say 20-something traits, and it all deals with facial traits, whether it's dimples or the shape of the eyebrows, or the shape of the ears, or the shape of the forehead, or shape of the chin. There's so many different traits, and what y'all do, you first establish who's mom and who's dad, which would be easy in y'all's case, I would hope. And if not, then like y'all would flip a coin, who's mom, who's dad. Then you'd flip a coin to see what sex the baby's going to be. And then you flip a coin to see what you're going to name it. You know, you have to agree on a name. We have to name the baby. And then we'll start flipping a coin per trait. So mom will flip it and write down her one component. Dad will flip it and write down that. And then it'll show you on the paper that gives you this chin shape or this nose shape or this. And when it's all over, you draw all these shapes out. So you'll want to bring colored pencils. Bring colored pencils, and we'll draw these out, and you'll see some good looking kids, and you'll see some of the ugliest kids you ever seen in your life.
We did this in my advanced biology class in high school, and then we had to hang them out in the hallway for everybody to see.
We won't do that.
Those are some ugly kids.
We'll just make fun of them in here.
Yeah, there's some funny looking kids.
Well, I've seen some that literally only a mother could love. It was really sad. We had unibrows and everything else. And there's like 8 different genes involved in skin pigment too. I mean, it went from dark brown to snow white and everything in between. So just flipping those coins and hitting those, and that's what, and it really teaches you what genetics is all about. It's random chance. I mean, you got the same gene, everybody in here will have the same gene pool to work from. And not one of your kids will look alike. Not one of them. That's crazy. But we'll try to do that. We have a lot of fun with it. And Lord, I hope y'all are good artists, because sometimes that tends to be the problem. But they do give you what the shapes look like on the paper so we can put it together. We'll try to do that. All right, moving on over to monohybrid. That's a 1-trait cross. We'll do some of those today before we quit. A dihybrid is a 2-trait cross. We'll probably do those next Thursday. And then a test cross is a cross between a unknown genetic makeup, a plant, in Mendel's case, it's a plant with unknown genetics. We cross it with a homozygous recessive. So, unknown crossed with homozygous recessive. Why don't we cross it with a recessive? Because the recessive has nothing dominant to mask anything of the unknown plant. We cross it with the recessive to express the unknown plant's genetics so we can figure out what the unknown plant is. So what Mendel did, and this is how he discovered this process, you can already probably imagine just how meticulous he was in what he did. He probably had a pretty good sized garden, had a lot of space between his plants. He may have his dwarf plants over here and his real tall ones way over there and a lot of space. And out here in the middle of his garden, I can just see this volunteer pea plant coming up. And something like that absolutely drove him nuts, because he knows it's a pea plant, it's in the middle of his garden, he did not plant it there. Is it tall? He's thinking genetics. Is it homozygous tall? Is it heterozygous tall? Is it dwarf? Well, we can solve that pretty quick. Well, nothing happens quick in plants. You ever sit around and just watch a plant grow? Imagine how exciting his life was, because that's what he did. You let this plant grow up until it matures. If it's as tall as his tall plants, we know that it has a tall trait, but we don't know if it's pure or not. We don't know if it's homozygous tall or not. So what Mendel did, let's say it grew up tall, he went over to his dwarf plants and he gathered some pollen when it started flowering, and he pollinated this plant with dwarf plant pollen. That's homozygous recessive. And then he covered the plant up. Now, why would he cover the plant up? Was he ashamed of it? Was it an ugly plant? No. He covered it up to prevent what? Other pollination. He didn't want wind to blow tall pollination over there and mess up his, so he covered it up. Now, did that prevent an insect from crawling up in there maybe? And no, so there's still a possibility of some cross-pollination, but he limited that for sure. Rain and wind was out of the picture here. So now he let this plant go ahead and produce seeds. He let the seeds mature. There's a lot of time going on here. He's sitting over in a chair watching it, I'm sure. He takes these seeds, let's say he takes 50 seeds out here from this plant, and now he plants them. They germinate. They come up. He watches them grow. This is like taking a year, isn't it? Forever. And he notices that out of, and he's a green thumb, so every one of the seeds come up. So out of the 50 seeds that come up, he noticed that 47 of them were tall. 3 were dwarf. He already knew on paper that the unknown plant was either big T, little t, or big T, big T. Would you agree? We knew it was tall. So he crossed that with the dwarf. And he crossed this one with the dwarf. He already knew on paper what he was looking for. So what was the unknown plant? Was the unknown plant homozygous tall or heterozygous tall? Heterozygous tall? Now, if you look in theory here, if the unknown plant was heterozygous tall, we should be 50-50 tall and dwarf. Were we 50-50? This unknown plant was homozygous tall, because 47-3 ratio is much closer to being 100% than, let's say, 23 to 27. See what I'm saying? If it was 23 tall and 27 dwarf, then we got a 50-50. It's never going to come out perfect. There's always some bug's going to throw another pollen grain in there. What if the wind hit it before he got it pollinated and covered? There's all kinds of things that can come into play. Never comes out perfect in theory, but that's how he come up with a test cross. Always cross the, why didn't y'all tell me I spelled unknown wrong? The unknown, there we go, the unknown times homozygous recessive. Last two terms that we have to get a grasp on is dominant recessive. And we've already been talking about dominant recessive. Dominant is the expressed trait. It's the trait we can see. It's the one that's going to dominate and show up. It's the expressed trait. And recessive is the trait that is usually masked. When is it not masked? When is a recessive trait expressed? When it's in a homozygous form, when there's not anything else there to cover it up. That's right. It's in a homozygous form. So know these terms. We'll apply these terms as we go through this chapter. Look on the first page, if you will. It's page 72, chapter 5. There's quite a few things I'm going to point out as we go down through here. Here is genes. See that in bold print there? And, you know, they talk about it there for nearly a whole paragraph, but they don't give you a real definition until the next paragraph. The first sentence of the next paragraph is your definition. A gene is a unit entity of inheritance. A unit entity of inheritance. It's a small component of what you get from your parents. Would you agree that we've connected a lot of things to genes? Cancer, hair color, eye color. I mean, we've got a lot of it nailed down. And they're all time finding something else, something new that's connected. They're coming out with a new kind of cancer or something else that's created here. But one of the latest things they've connected here, they claim that diarrhea is genetically inherited. Would y'all agree with that? If you think about it, it runs in your genes, don't it? That was my joke of the day. Not a good joke, huh? It's pretty pathetic. Yeah, it runs in your genes, so it depends on how you look at it, I guess. But it's a unit entity of inheritance. Mendel's investigations, this talks about his history here and the things he studied and the years he did it. And he even talks about him being the father of genetics there. Go over to the right-hand side in the term gamete. These are special germ cells or gametes. They'll reference these as being alleles or gametes a lot of times. And for every trait you have, you'll have two gametes or two alleles. One from mom, one from dad. Okay? You don't get two from mom, you don't get two from dad, or dads don't beat moms up and take over. You get one from each. And that's what they're going to reference that quite a bit. Now, if you're missing one, or if you have one too many, then those are mutations. And we'll talk about those as we go through this. Now, there's meiosis. We just covered that, so that's good. We don't have to go into that. And it takes us all the way over to page 75. Page 75 talks about sex determination. Well, in humans, I think we understand that XX is a female in humans, and XY is male in humans. But that doesn't hold true in the whole animal kingdom across the board. You can flip-flop that because in birds, butterflies, and moths, XX gives you a male. That's in birds, butterflies, and moths. And then XY gives you a female in birds, butterflies, and moths. Why would that be an advantage to birds? We're more familiar with birds probably than butterflies and moths. How could that be an advantage? Do you know what a bird is?
So is that like why the male cardinals, like they're red? Like you'd think since they're prettier it'd be the female bird. I mean, you know what I'm saying?
I think you're assuming that in humans females are prettier than males, and that hurts my feelings. That's a good observation because
live shorter lives and reproduce more.
birds do live shorter lives and they do need to reproduce, but I don't think color, has a lot to do with that. Color in birds is sex linked. And for the male, which is the prettiest in birds, for the male to get the combination of colors that they do, they have to have both X chromosomes to carry all those combinations of colors. If they just have one X, then they get that little drab camouflage looking, just okay kind of color, you know. So the males need this because they're gonna create a territory. Birds are extremely territorial. If we get to that chapter, you'll see why. Birds are extremely territorial. So the male's gonna establish a territory. They're gonna fight tooth and toenail. There's gonna be a pecking order. Somebody's gonna get beat up. Somebody's gonna get the best territory. And the best territory is gonna provide everything that the female needs. Ultimately, the females choose their male. It's not like the pretty males are picking among drab females. The females are still picking pretty, but they're also picking dominant. They want the one that can establish a territory that has food, that has shelter, has all the qualities that they need to raise young. And so the males with the color, they can be seen. They like to show off, they like to make noise, they like to be seen. Have you ever seen, talking about redbirds, you ever seen one of those fly by your window and make a U-turn, come in and just peck your window to death? You ever been woke up by birds pecking on your window in the springtime? Y'all are heavy sleepers. What they do is they fly by the window and they saw a reflection of themselves in the window, and in spring, that's when they're setting up the pecking order, and they think, there's another male in my territory. So they come back and fight themselves in the window. Now you're talking about bird brains here, and they were not very smart. Of course, I've seen them run into sliding glass doors and kill themselves too. Of course, I've run into sliding glass doors. I lived through it. But. So in birds, butterflies, moths, it's the color that's connected here. And sex determination, it's different in some animals than it is in humans. See if there's something I missed there. I don't think so. All right, move over to 76. When we get into a lot of the terms we just put up on the board and some of Mendel's laws. And his first law states that in the formation of gametes, paired factors that may specify alternative phenotypes separate so that each genotype, or each gamete, receives only one member of the pair. In other words, if mom offers big T, little t, the child will get one of those. It's random assortment. One child may get this one, next child may get this one. We don't know. All the children may get this one. That's why we do the Punnett square. We see what the chances of that being. If mom has these two big T's, the child's only going to get two big T's. So when we start working with dihybrid problems, let's say big T, big Y, little y, Mendel's pea plants here, we have to look and see all the different combinations that possibly could happen here. We could have a big T, big Y combination. We could have a big T, little y combination. We could have a little t, big Y, and a little t, little y combination. There's 4 different combinations you could get from those 2 traits that mom could pass on to the offspring. So all he's saying is random. We don't know which one's going with which one. And every one will be different. When we draw our pictures of our kids, when we do that extra, you'll know. Everybody in here's got the same gene pool. None of you have the same looking kid. I've never seen it yet. It's pretty amazing. Now that's just facial features. Start throwing in all the other traits that we actually deal with on a daily basis. All right, test cross is listed over on 77. There's also a section in here, a short section on what's called probability. What's the chances of something happening? And now we're already saying it's pretty slim. I mean, how many of y'all have a brother or a sister, have siblings? We asked that the other day, and they don't look anything like you, or they sure may not act. They may look more like you, but certainly don't act like you, and you have so many different variabilities. Well, that's what the law of probability is basically saying. The chances of it happening is very, very slim. So using the same gene pool, producing Twinkies all the time. It's just not likely. So what if, just use a simple example, what if, let's say this man and woman has had 10 kids, and all 10 kids was girls, but dad talked mom into trying one more time for a boy. What's the chances of that 11th child being a boy?
Not very good.
You think their chances of shot at this point? Actually, it's the same chance as if they'd never had a kid. It's still 50-50. Every one of those had a 50-50 chance of being a boy or girl. You just got X and a Y, right? So ultimately, probability really doesn't remember anything that's happened in the past. It's what's happening now. So I've created my own law here. It's called Dwann's Law. Y'all aren't wanting to write this down? I'm going to tell you what Dwann's Law is. This is important. Dwann's Law, you pick that pencil back up. Dwann's Law says, I can't believe y'all aren't writing this down. I made this up years ago. It's not in the book, but it will be one of these days.
We're on it, here we go.
Are you ready?
Are you gonna give us part of the cut when you make $1 million off of it?
No.
Okay, then I'm not writing it down.
This is going to come back to haunt y'all. Dwann's Law says that chance has no memory. Think about that. Pretty deep, isn't it? Every time you gamble, that remember you already lost $100,000?
Nope.
It's just going to take another dollar.
Wow.
Gamble again, did it know that you just won 25,000? No, it's planning on getting that back. So it has no memory of what has happened in the past. It's all right now. And the odds are against you, especially when you go to the casino. All right, moving on. I can tell you really didn't get excited about Dwann's Law. Let's look at some basic monohybrid crosses. Mendel had some set ratios that he liked to deal with, and Mendelian genetics became, and is today, very predictable. Now, predictable means when Mendel would see a particular cross, he already knew what was going to happen. He didn't have to work his square anymore. He could predict it. And hopefully y'all will get good enough to see some of that happening before we get finished with some problems in the lab. Let me give you an idea of some genetics that's going on today that's not predictable. We're using genetics for a lot of different things. Of course, in plants, we're making plants more resistant. We can create a single rose bush that'll produce every color rose that there is out there. Now, we have to go back to the beginning to produce that same rose bush. We can't take a clipping off that rose bush and produce that rose bush again, because if we take a clipping off that rose bush, we'll produce probably a single color flower again. So there's some things that we kind of force feed or force make, but you know, that's not predictable stuff. Here's a good example. I went back somewhere around 2000, I went to a seminar. It was up around Tulsa, but the guy speaking at it was a geneticist from OSU. He had worked for years at Texas Tech and had just recently taken a job at OSU. And he was talking about a lot of things that they apply in their genetics. Of course, those guys that's got doctorates in genetics, they teach 1 or 2 classes and spend a lot of time in research. And when he was at Texas Tech, his thing that he researched was a rare bat that lived in Australia. It wasn't the average bat you see around here. There wasn't even many of them in Australia, but he knew where they was at. So he spent his summers in Australia studying that bat. Come back here, had a few bats here, do research on them. When he was at Texas Tech, that's big cotton country out there, there were some farmers come to him and explained to him that 2 or 3 of these farmers owned a small seed company. Not big, but it was small. They survived with it. They had developed a strain of cotton that must have been second to none. It must have been a really good strain of cotton. And they had made pretty good money selling this cotton seed for several years, until the competition, the big seed company, all of a sudden had that seed. Underpriced them, made the seed company lose a lot of money. So they come to this geneticist and ask him, how can we, if we develop another one, how can we patent it to be ours? And then prove it when they steal it. When we take them to court and sue them over it, we got to be able to prove it. So this geneticist and a couple others kind of put their heads together and they thought, well, we're going to have a gene marker somewhere. There's got to be, you can't take every little seed and stamp it because it germinates, it's gone and all that. So there's got to be a genetic marker. So what he did is took a gene from this rare bat he studied and implanted it in the plant genetics. They found a place in the genetics. See, we have a lot of DNA that's used. We have a lot of DNA that does nothing. And that's what we went into the DNA area of this plant that they felt like didn't play a role, if any role, into this plant, and they stuck this gene in there. But every time the cell replicates, it'll replicate the stuff that's used and the stuff that's not. So he estimated that if that gene went through 5 generations of that plant plant would keep it forever. They felt confident with 5 generations. After the 3rd generation, they couldn't find the gene again. The plant had kicked it out. So they went back in and found another place and put the gene in again in another place. To get it to stay for 3 years, they thought, we've got something going here. They put it in another place, and he left after the 4th year. He left his comrades with the rest of it, but he said after 4 years it was still there. So he felt confident that they were going to be able to do this. So here we have a cotton plant that has a rare bat gene in it. And if somebody were to steal that seed and say, oh no, it's just, this is a different kind, and they go plant it, well, they could take that cotton plant and look in the genetics of it, find that bat gene and say, that's my bat gene. It's not just any bat gene, that's my bat gene. And we patent it by the bat gene and where it's at. They could prove it in court and they'd hold up. So that's non-predictable. I mean, obviously, well, just think about it. Ultimately, we could have a cotton plant that would pick itself at night and fly into the gin. That'd be pretty cool, wouldn't it? That'd save on some labor, wouldn't it? That's non-predictable stuff. Well, Mendel's stuff is predictable. And just some of his simple stuff, we already have T is tall, little t is dwarf. And he always liked to cross 2 homozygous, big T, crossed with little t, little t. Now, we can see the obvious here. This parent's only going to offer the big T, correct? And this parent's only going to offer the little t, so all the offspring is big T, little t. All the offspring are tall, but they're heterozygous tall from 2 homozygous parents. Homozygous recessive, homozygous dominant. He calls this his F1 generation, and then he likes to take his F2, this one, and cross it. Big T, little t, big T, little t. And F stands for filial. First filial generation, second filial generation. So when he did this, he started coming up with some predictable ratios. Big T, little t, big T, little t. He saw that the first one was homozygous dominant, second one was heterozygous like the parents, and the third one was total opposite, homozygous recessive. So Mendel had some set ratios that he went by here. Every time in a monohybrid cross, if he crossed 2 heterozygous, 2 heterozygous parents, he would always get a phenotype. What's phenotype again? The physical. He'd always get a phenotype of 3 to 1, 3 tall to 1 dwarf. And he'd always get a genotype of 1, 2 to 1. And that's represented by 1 big T, the 2 big T, little t's, and 1 little t, little t. That became his predictable genetics. So when we get into dihybrids, we'll be able to predict dihybrids as well when you cross 2 heterozygous parents. And dihybrids, you end up with a 9:3:3:1 ratio, which is there's 16 squares in a dihybrid. That's the phenotype. The genotype in a dihybrid is 1, 2, 1, 2, 4, 2, 1, 2, 1. But we'll see how that plays out as we work that Punnett square. So, predictable, every time you cross 2 heterozygous parents in a monohybrid cross, predictable 3 to 1 pheno and geno is 2 to 1. Those will be test questions on test 2. I'll ask you those. I'll also ask you the 9-3-3-1, but I won't ask you the 1-2-1-2. You'll just have to work it in here. Let's look at another one. Let's say we cross a big T, little t with 2 little t's, kind of like we did with this test cross a while ago. What we get? What's our phenotypic ratio?
It's gonna be 3-1.
What's our phenotypic ratio? Physically? Are this one tall? This one tall? Dwarf? Yeah, 2 to 2, or you could even condense it to 1 to 1. All right, what about genotypic ratio? Still the same. Yeah. Genetically, we have a big T, little t, and a little t, little t. So it could be 2 to 2 or 1 to 1 again. So as long as, again, understanding what the term genotype and phenotype means, if I'm asking for phenotype, you look at it, find the dominant trait, and say, that one's tall, that one's tall, that one's tall. That's what they're physically going to be like. And genetically, you look for the ones that have the exact same alleles. Only one of those. It's 2 of those, 1 of those. Does that make sense? Genotypic, phenotypic. Are y'all alive? Hanging in there? Okay, so there's some basic monohybrid problems. On page 78, Mendel gets into his codominance. We mentioned a little bit of this a while ago with the 4 o'clock flowers is what Mendel worked with. Of course, in zoology, they have to use something different. They have to use an animal, right? So they used blue undulation chickens. And there's 3 terms here. And there'll be the second set of problems that I'll give you to work, probably week after next. In that second set of problems, there'll be a question in there that you'll have to recognize the fact that this is an incomplete dominant situation. So this is where you have 2 colors that's not dominant to each other. The 2 colors come together and blend and give you a new color. So the white one and the red one gave you pink one in the flowers. So the terms are intermediate inheritance, codominance, or incomplete dominance. It all means the same thing. I think they use at least 2 of them in this book. So here they're using blue undulation chickens. Come up with this by accident, like most things. They crossed a white speckled chicken and a black chicken, and the offspring were all blue chickens. Slate blue. Absolutely, as far as chickens go, pretty. Now, they thought, boy, a pretty blue chicken. We can sell pretty blue chickens. So let's produce more pretty blue chickens. So it only makes sense to cross blue chickens with blue chickens. You should get blue chickens? No. When you cross a blue chicken and a blue chicken, 50% of them were blue chickens, but 25% of them were white speckled chickens and 25% black chickens. So they was doing all kinds of mixes and matches here. Well, okay, let's cross a blue chicken and a black chicken. Well, you got 50% blue, 50% black. Let's cross a blue chicken and a white speckled chicken. 50% blue, 50% white speckled. If we cross 2 blue chickens, obviously 50% blue, 25% white speckled, 25% black. Only time you ever get 100% blue is when you cross the black chicken and the white speckled chicken. You get 100% blue. It took me years to get that little lecture down. I usually do it a little bit faster. I tried to articulate it better. But that is incomplete dominance. And, one obvious case to me out in the animal world of this, if you're familiar with cattle, Black Angus and Charolais. Anybody know what color Charolais is? It's as white as it gets. What do you get when you cross those 2? Doesn't matter if it's a black bull and a white cow or a white bull and a black cow. Doesn't matter. What do you get? A blue cow. A what cow? A blue? A blue cow. That's a good guess. I hadn't thought about that. You get a smoke-colored calf. In bovine and beef, black is a very dominant color and white is a very dominant color. You mix the 2, they don't dominate each other. You get this smoky colored calf. So that's an incomplete dominance. Okay. That's just an obvious one that I see out there. All right, let me work a couple of these with Mendel's 4 o'clock flowers. let's say we'll just do R is red, W is white, and RW, of course, it had to be WW and RR. RW would be pink. There's your combinations. So, like the blue Andalusian chickens, what happens if you cross two pinks? What are you going to get? You get two pinks, a red, and a white. 25% red, 25% white, 50% pink. What happens if you cross a red and a white? 100% pink. What happens if you cross a pink and a white? 50-50. Pink and a red? 50-50. Very good. No dominance, but we tend to revert back a lot with it. If you want to get 100% pink, you got to go back to the original parents to get 100% pink. We hanging in there so far? What time is it?
12:30.
12:30. No. I hear you. I didn't even bring lunch today. I thought I'm just going to work off 5 pounds in genetics. Let's last little deal I want to do today, and we'll be out of here before 1. Let's do some sex-linked problems, and we touched on that just a minute while ago. But in sex-linked, we have XX gives you a female, XY gives you a male. And if we're working with color blindness, like we did a while ago, that would be a colorblind female. This would be a carrier female. In other words, physically, phenotypically, she sees fine. But genetically, she's carrying that colorblind trait. And then you have the colorblind male. If he has the one colorblind trait on the only X he has, he's colorblind. Now, let me tell you a little bit about the Y. The reason these traits tend to be carried on the X is because the X is much bigger than the Y. The Y is little. I mean, in all reality, we should be writing an X up here like this, and the Y about like that. Is the Y significant? If you want to be a boy, it is. Is the X significant? If you want to be alive, it is. If you don't have an X, you're dead. You have to have an X to be alive. So that's significant. But in sex-linked information, and I'll probably ask some kind of test question about this, if you hear the terminology sex-linked, where is it carried most of the time? On the X, right? Now, is there any sex-linked information on the Y? Yes. There's, first of all, there's what's called a testes determining factor. That's on the Y. That's why the Y makes a boy. The testes determining factor is there, turned on. Then testes develop in embryonic development. The right hormones come about, we develop a male. And in fact, for a female to be developed, and this talks about this in chapter 7, which is on our finals, but I always touch on it in genetics. There's a 10-day time frame that in embryonic development, we wait on the testes determining factor to be turned on and testes to develop and testosterone to be produced. There's a 10-day time frame from when a male starts developing, if that's there, to when a female starts developing, if it's not there. In chapter 7, don't throw anything at me, but I'm just quoting chapter 7. It says the female is the default to the male. And all it's saying is, if the testes determining factor is not there, then they'll go ahead and produce a female. If it's there, we obviously have a male. That's all. But it delays itself to make sure we don't screw up anything in the process. We want to make sure that that factor's not there. So there's a 10-day time frame in developmental stages. All right, now, with that said, the only other thing carried on the Y is a trait called hairy ears in men. Isn't that exciting? Now, this never misses a generation. If a man has hairy ears, his sons will have hairy ears, and their sons will have hairy ears, and their sons, it doesn't miss. what does dad give his son? The Y. If it's on the Y chromosome, he gave him hairy ears. Now, how many of you ladies are already married? That's too late for y'all. The rest of you, if you're not married, or if you're dating or in a relationship, go look at the guy's dad. You know, if you like him, you may give him a hug, peek in his ear. If hair stabs you in the eye, get out while you can get out. Get out while you can run. Run while you can. But unless you want sons with hairy ears, there's your choices. Now, it'll take me just a minute. Been thinking about it, I want to brought this up.
We're just gonna take the nose hair trim into your ear. No big deal.
I think I've got it on here somewhere.
Hairy ears?
No, I'll have to go over there. Are we up and running up there yet?
No.
No.
Is it counting down? Can you see anything? Yeah. Yeah, I see it.
It's trying.
As you can see, Radicant Badge Pie's ear tufts measure almost 25 centimetres, beating the previous record holder by a good 10 centimetres. He's a grocer from Uttar Pradesh, and he's made it into the Guinness World Book of Records. Coming up,
People, that's
Why?
hairy
That's gross.
ears. You know what he does for a profession?
Braids them? What?
He's in the food industry.
Yeah.
Hair nets don't cover his hairy ears. I've seen another video clip of him going down food aisles, checking out food. He's got a hairnet on, his hairy ears are just... And they ship their food over here to us. Isn't that nice?
Spiders on drugs.
I'll come back to that in just a second.
Oh, that's so gross.
But I had to show you that, so you can get a visual of what you're looking for, so you can run while you can.
I hope he doesn't have children. I pray he doesn't.
Actually, I think he has a son.
Oh, that poor child.
Yeah. Guess what his son has?
Hairy ears.
Yeah, y'all are good. Y'all are really catching on the genetics today.
Oh.
All right, let's work a couple of these color blindness, and I'll say this too. However these colorblind problems work out, hemophilia, the bleeding disease of King's, you ever heard of it?
I have it.
You have it? It also works exactly the same way genetically. So if you have it, then you would have it on both X chromosomes. So, all right, so let's just take, let's say mom is a carrier, and let's say dad is not. We work the Punnett square. We have a daughter that's a carrier. We have a daughter that's not. We have a colorblind son and a normal son. So if I ask the question, how many colorblind sons would you get? One. 50% of the sons would be colorblind. How many colorblind children? 25%, right? Yeah, because she's not colorblind. So be careful when you read a question that you read it as if it's asking for how many females, how many sons, or how many children. So if it's females or sons, daughters or sons, then you're either gonna be none, or 100%, or 50%. And if it's children as a whole, you could have 25%, 50%, 75%. See what I'm saying? Well, be sure you read that question correctly. All right, now, if we take that out, we already did one while ago where mom was a carrier and dad's colorblind, and dad got the blame for it, but we know because of our genetic background now, right, that mom is the one to pass that on. So let's do one here that dad's the colorblind one, and he would provide 2 daughters to be carriers. We still wouldn't see colorblindness in this generation. What we'd see is possible colorblind grandsons, wouldn't we? In the next generation, mom would have a 50% chance of producing colorblind sons. Now, what if she had 2 sons? Could they both be colorblind? Sure. I mean, the odds are one of them will, one of them won't, but could they both be normal? Sure. So let's look at a situation like, do the last example right here. We're gonna have a colorblind mom. That's my grandma. My grandmother passed away a few years ago. She's colorblind. And she had 3 sons. What's the chances of a colorblind mother and a normal father producing normal children? What? She only has colorblind X's to give her sons. What's all of her sons gonna be? They're all gonna be colorblind. The daughters, all carriers. My family tree on that side's gotten out of control. There's too many grandkids and great-grandkids out there now. But I do know my dad and my 2 uncles are colorblind. And I do know I have a couple of cousins out there that's colorblind. And I do know my 2 aunts have to be carriers because of this combination right here. So, out of 5 children, my grandmother produced 3 colorblind boys and 2 carrier daughters. And the carrier daughter is going to keep producing. And you know, I think there's that 1 great-grandchild out there that they've identified as being colorblind.
So, if I were to have a boy, since I am hemophiliac, I'd end up giving him.
He would be a hemophiliac. Yep.
And if I had a daughter, she'd be a carrier.
She'd be a carrier.
Okay.
She would be normal, but she'd be a carrier. For a chance for her, 50% chance of her sons, but she would not have a daughter that's hemophiliac unless she marries somebody that is hemophiliac. Yeah. So this is a pretty simple deal to look at and understand for sure. So kind of gives you an overall idea of sex linked. If you hear sex linked, it's carried on the X with only a couple carried on the Y, right?
I know my cousin has cystic fibrosis.
Yeah.
When I was pregnant with my daughter, they tested me to see if I was a carrier of it.
Yeah.
Since it was my first cousin.
I'm not sure what gene that's carried on, what chromosome set that's carried on. It's not sex linked, but.
No, I just know that they said that my aunt was a carrier and my uncle was a carrier, and I think my cousin was. And they said if they had another kid, that it was a high chance that it would be cystic fibrosis too.
Well, every one of them runs the same chance, but if you don't mind, since we're finished, turn that off. I want to do a couple more things.